In a Wyoming winter in 1981, a ranch dog named Shep trotted home and dropped a dead animal on his owner's porch. The rancher had never seen anything like it. Slender body, a black mask across its eyes, black feet. He took it to be identified, and the answer set off a quiet earthquake in the world of conservation. The animal was a black-footed ferret, a species that scientists had already declared extinct. It was not supposed to exist anymore. And yet here was one, dead on a porch, which meant somewhere out on that ranch, there were more. That single dead ferret led scientists to a hidden colony of survivors, the last black-footed ferrets on Earth. But the rescue that followed would run into a problem that no amount of careful breeding could fix, a problem buried deep in the animal's DNA. And the solution, when it finally came, would involve reaching back in time, into a freezer where the cells of a ferret that died in 1988 had been waiting in suspended animation for over 30 years. What scientists did with those frozen cells had never been done before with an endangered American species. This is that story. Let's begin. The black-footed ferret was once an animal of the open American West, and its entire existence was tied to a single creature, the prairie dog. The black-footed ferret is North America's only native ferret, a slim, masked predator built to slip down into prairie dog burrows and hunt the rodents in the dark. Prairie dogs were its food, its shelter, and its whole world. The ferret depended on them so completely that the two species were effectively bound together. Where the prairie dogs thrived, the ferrets thrived. And when the prairie dogs began to vanish, the ferrets were doomed to follow. The collapse of the prairie dogs came at the hands of farmers and ranchers across the 20th century. Seeing prairie dogs as pests that competed with cattle and pocked the land with burrows, people poisoned and exterminated them across the Great Plains on a massive scale. As the vast prairie dog towns were wiped out, the black-footed ferrets that depended on them starved and disappeared along with their prey. By the 1970s, the black-footed ferret had become so rare that scientists concluded it was gone. The species was officially considered extinct, another casualty of a changing continent. And then came Shep the ranch dog, and that single carcass on a Wyoming porch in 1981. When scientists confirmed what the dog had found, they raced to the ranch and discovered a small surviving colony of black-footed ferrets clinging to existence, a population that had somehow hung on, unseen, after the species had been written off as extinct. It was a miraculous second chance. But the celebration would not last. Within a few years, disease swept through that last wild colony, and the ferrets began dying fast. Facing the total loss of the species, scientists made a desperate decision in the mid-1980s. They captured the last surviving black-footed ferrets out of the wild, every one they could find, to start an emergency captive breeding program. It was the same agonizing gamble made for the California condor in the same era. Take the final animals out of nature entirely, accept that the species would vanish from the wild, and bet everything on breeding them in human care. When the capturing was done, the entire future of the black-footed ferret rested on an astonishingly small number of animals. Just 7 of them would end up successfully reproducing. 7 founders. That was the entire genetic foundation of the species. That number, 7, is the hidden problem at the heart of this whole story. Every black-footed ferret born in the decades that followed, every single one of the hundreds bred in captivity and released back into the wild, descended from just those 7 founding animals. When a whole species is rebuilt from 7 individuals, it carries a curse known as a genetic bottleneck. The gene pool becomes desperately shallow. The animals are all, in effect, close relatives, and that lack of genetic diversity is dangerous. It makes a species more vulnerable to disease, less fertile, and less able to adapt. The black-footed ferret had been saved from extinction, but it had been saved into a genetic trap. For decades, the breeding program fought hard against that trap and achieved something remarkable. Through careful, intensive captive breeding, the black-footed ferret population was rebuilt from those 7 founders into the hundreds, and the animals were reintroduced across 8 states in the Great Plains. It was one of the great comeback stories in American conservation. But underneath the success, the bottleneck never went away. Every new ferret was still drawing from the same 7-founder gene pool, and threats like sylvatic plague and canine distemper kept exploiting that genetic weakness. To truly secure the species, the scientists needed something they did not have. They needed new genes. They needed an eighth founder. And there were none left to find. Or so it seemed. The answer to that impossible problem had been sitting frozen in California the entire time, waiting. To understand it, we have to go back to a decision made decades earlier by a geneticist named Oliver Ryder and his colleagues at the San Diego Zoo. Back in the 1980s, Ryder and other visionary scientists had begun building something extraordinary called the Frozen Zoo, a vast cryogenic bank of preserved cells and tissues from rare and endangered animals. Their reasoning was simple and far-sighted. They could not know what future technology might make possible, so they would preserve the raw genetic material of vanishing species now, banking it against a future where it might be needed. It was an act of pure foresight, saving something whose value would only be understood years later. In 1988, into that frozen zoo came the cells of a wild black-footed ferret named Willa. Willa was one of the last wild ferrets, and when she died in 1988, she died without ever reproducing. She had no descendants. In the normal course of things, that would have meant her unique genetics were lost forever, gone from the species the moment she died. But because tissue samples from Willa had been sent to San Diego and frozen, her genetic material did not vanish. It was placed in cryogenic storage, cooled to preserve it indefinitely, and there it sat, untouched, year after year, a single thread of a vanished animal held in suspended animation. For more than 30 years, Willa's frozen cells waited, and during that time the science quietly caught up to the dream. Then, in 2014, a genomic study of Willa's preserved DNA revealed something that made her frozen cells suddenly priceless. Willa's genome held nearly 3 times more genetic diversity than the entire living black-footed ferret population. Think about what that means. A single ferret who died in 1988, who never had a single offspring, carried in her cells the lost genetic variation that the whole modern species was missing. She was, genetically, the 8th founder the scientists so desperately needed. The only problem was that she had been dead for over 3 decades. There was only one way to bring her genes into the population. They would have to clone her. The plan to clone Willa brought together a powerful coalition. The U.S. Fish and Wildlife Service, the conservation nonprofit Revive and Restore, the San Diego Zoo Wildlife Alliance, and a company called Viagen Pets and Equine, which had spent years refining the technology of cloning domestic animals, all joined forces. The process they would use was the same fundamental technique that had created Dolly the sheep decades earlier. They would take a cell from Willa's frozen tissue, extract its nucleus containing her complete genetic code, and insert it into an egg cell from a common domestic ferret, an egg that had had its own genetic material removed. The egg would then be coaxed to begin dividing as if it had just been fertilized, growing into an embryo that was, genetically, a perfect copy of Willa. On December 10th, 2020, the impossible was born. A black-footed ferret kit came into the world, a living, breathing genetic twin of Willa, the ferret who had died more than 30 years before. They named her Elizabeth Ann. She was the first clone of any endangered species native to the United States. A healthy animal carrying the resurrected genetics of an individual who had been dead for over 3 decades. The cells that had waited in a freezer since 1988 had become a living animal again. It was a genuine scientific landmark, a moment where conservation and science fiction collided and produced something real. If a story about scientists reaching into a freezer to resurrect a dead animal's genes is the kind of thing you want more of, take a second and subscribe. Because here is where the story takes a harder turn. Elizabeth Ann was alive and healthy, but she could not do the one thing the entire project depended on. She could not breed. When Elizabeth Ann reached maturity, scientists discovered she had developed a condition called hydrometria, a reproductive problem that made it impossible for her to carry offspring. She had to undergo surgery, and though she recovered and remained healthy, she would never pass on Willa's precious genes. The first clone, the historic animal, the proof that it could be done, turned out to be a genetic dead end of her own. The breakthrough had happened, but the actual goal, getting Willa's diversity back into the living population, was still out of reach. The scientists did not stop. They went back to Willa's same frozen cell line and cloned her again. In 2023, two more genetic twins of Willa were born, named Noreen and Antonia. These were Elizabeth Ann's identical clones, carrying the exact same resurrected genome. Noreen was raised at the National Black-Footed Ferret Conservation Center in Colorado, and Antonia at the Smithsonian's National Zoo and Conservation Biology Institute in Virginia. This time, the hope rested on them. If even one of these cloned ferrets could successfully breed, it would do what no clone of an endangered species had ever done. It would inject brand new, decades-old genetic material into a living population and break the bottleneck that had constrained the species for 40 years. In June of 2024, it finally happened. Antonia, the cloned ferret carrying Willa's resurrected DNA, was bred with a wild-type male named Urchin, and she gave birth to healthy kits. It was the first time in history that a cloned, endangered animal had ever produced offspring. The moment those kits were born, Willa's genes, frozen since 1988, lost for over 30 years, finally flowed into the next generation of living black-footed ferrets. A ferret who had died without descendants in the 1980s had, through cloning, become a mother to a new lineage in the 2020s. The eighth founder had arrived 36 years late. The impact of that birth reached far beyond two kits. For 40 years, every breeding ferret in the program had descended from just 7 founders, and Willa's genes had been completely absent, 0% of the population. By bringing Antonia's offspring into the gene pool, scientists officially established Willa as the 8th founder of the entire species, the first new genetic line added in 4 decades. Within a few years of Elizabeth Ann's birth, more than a dozen ferrets in the breeding population carried Willa's genetic material. The bottleneck that had locked the black-footed ferret into a shallow, dangerous gene pool since the 1980s had finally, genuinely been broken, not with a new discovery in the wild, but with a frozen cell and a cloning needle. But this story does not end in a clean and simple triumph, and it would be wrong to tell it that way. The 2 clones who came after Elizabeth Ann did not all live to see the full results of their importance. In 2025, both Elizabeth Ann and Noreen passed away. Elizabeth Ann, the historic first, never bred at all. The miracle of the program rested almost entirely on Antonia and her offspring, on the narrow success of a single cloned animal managing to reproduce. The technology had worked, but it remained fragile, dependent on a tiny number of animals and an enormous amount of human effort, money, and luck. Cloning had cracked the bottleneck open, but it had not made the black-footed ferret safe, and the deeper threats are all still out there, waiting. The black-footed ferret is still one of the most endangered mammals in North America. The prairie dogs it depends on are still under pressure, still poisoned and reduced across the plains. Sylvatic plague still sweeps through ferret populations and can wipe out entire colonies. The cloning of Willa added precious genetic diversity, but it did not restore the prairie, did not cure the plague, and did not remove the fundamental fragility of a species that humans nearly erased. As the scientists themselves are careful to say, cloning complements the hard work of habitat protection and disease management. It does not replace it. The freezer gave the ferret a fighting chance, not a guarantee. Step back, and the full story of the black-footed ferret becomes one of the strangest and most hopeful in modern conservation. A species declared extinct was rediscovered by a ranch dog, rescued down to 7 founders, and rebuilt into the hundreds, only to be trapped by the shallow genetics of its own narrow survival. And then, decades later, it was rescued a second time by the foresight of scientists who had frozen the cells of a dead ferret named Willa in 1988 without knowing exactly why, only that they might one day be needed. They were needed. 36 years later, those frozen cells became living, breeding animals, and Willa finally took her place in the species she had left behind. What does all of this mean for the black-footed ferrets slipping through the prairie grass today, hunting in the dark down prairie dog burrows? It means some of them now carry the genes of an animal that died before they were born. An ancestor reached out of a freezer and dropped back into the river of life decades after her death. The black-footed ferret did not just survive extinction once. It was pulled back from the edge twice. The second time by a technology its rescuers in the 1980s could only have dreamed of. Willa died without descendants. And yet today, her bloodline runs again through the wild grasslands of the American West. Proof that sometimes what is frozen and forgotten is not truly lost, only waiting. If this story stayed with you, subscribe. There are more secrets still frozen and buried in the DNA of the animals all around us, and we are only beginning to uncover what they can do.